Bismillahirrahmanirrahim. The control of male sexual function by hormones is discussed in this video. Male sexual functions are controlled by hypothalamus and anterior pituitary gland. The hypothalamus secretes GnRH, that is gonadotropin releasing hormone, in a pulsatile manner, and this GnRH travels via the hypothalamic hypophyseal portal circulation to the anterior pituitary gland. And here, this GnRH stimulates the anterior pituitary to release gonadotropic hormones that are the luteinizing hormone and follicle stimulating hormones. The luteinizing hormone and follicle stimulating hormone, that are the hormones that are secreted from anterior pituitary under the influence of gonadotropin releasing hormone. Then uh, these among these, this luteinizing hormone, it primarily acts on Leydig cell in the testes and stimulate the secretion of the testosterone. And this testosterone is the key hormone for development of male secondary sexual characteristics that are voice, muscle mass, and hair distribution. Also, they are responsible for maintenance of libido and support of spermatogenesis, while this follicle stimulating hormone, it acts on the Sertoli cells of the seminiferous tubules, and its main function is to stimulate the spermatogenesis. The Sertoli cells also secrete inhibin, which exert negative feedback on follicle stimulating hormone secretion. Testosterone itself, it inhibits gonadotropin releasing hormone and luteinizing hormone. That is the negative feedback for both the hypothalamus and pituitary. And the inhibin from the Sertoli cell, it inhibits follicle stimulating hormone secretion at the pituitary. So hypothalamus, through the GnRH or gonadotropin releasing hormone, it causes the pituitary to release luteinizing hormone and follicle stimulating hormone. Luteinizing hormone acts on Leydig cell and causes release of testosterone, while follicle stimulating hormone acts on Sertoli cells and it is responsible for spermatogenesis, and also there is secretion of inhibin from the Sertoli cell that acts as negative feedback for the follicle stimulating hormone. So the feedback loops maintain hormonal balance and control male reproductive function. This illustration is describing the hormonal regulation of the male reproductive function through hypothalamic pituitary testicular axis. The hypothalamic, pituitary, and testicular axis. So the hypothalamus secretes gonadotropin releasing hormone, and this gonadotropin releasing hormone, it travels via the portal system to the anterior pituitary gland, and here, in response to the gonadotropin releasing hormone, there is release of luteinizing hormone and follicle stimulating hormone. The luteinizing hormone acts on the Leydig cell in the testes and stimulate the secretion of testosterone, and testosterone then it is responsible for promotion of androgenic effect, or that means secondary sexual characteristic, libido and muscle mass, and it also provides feedback control to both to the hypothalamus and pituitary gland. follicle stimulating hormone, it acts on Sertoli cell and stimulates the process of spermatogenesis. Sertoli cell can also produce inhibin, and this inhibin, which specifically inhibit follicle stimulating hormone, here follicle stimulating hormone, this inhibin, and this is the negative feedback for the hormone secretion. So testosterone inhibit both the gonadotropin releasing hormone from the hypothalamus and luteinizing hormone secretion from the pituitary gland, while inhibin inhibits follicle stimulating hormone secretion at the level of the pituitary gland only. So testosterone also acts on central nervous system, influencing the male sexual behavior and libido. Gonadotropin releasing hormone causes increased secretion of luteinizing hormone and follicle stimulating hormone. This is a 10 amino acid peptide that is secreted by a specialized neuron whose cell body lies in the arcuate nucleus of the hypothalamus. The axon terminal for these neurons and in the median eminence, releasing gonadotropin releasing hormone into the hypophyseal portal circulation. So this gonadotropin releasing hormone travels via the portal blood system to the anterior pituitary gland. In the anterior pituitary, it binds to the receptor on gonadotropin stimulating secretion of the two gonadotropins, the luteinizing hormone and follicle stimulating hormone. The pattern of this secretion is pulsatile. It is released intermittently in pulses lasting for a few minutes every 1 to 3 hours, and the stimulus, the strength of the stimulus, it depends on the frequency of the pulses and amount of the gonadotropin releasing hormone in each pulse. So the luteinizing hormone secretion follows the gonadotropin secretion, and almost closely and cyclically the pulse for pulse, while follicle stimulating hormone secretion, it responds less acutely to each pulse of the gonadotropin releasing hormone. And it fluctuates more gradually or hours in response to long-term changes in gonadotropin releasing hormone. Because of the stronger association between the gonadotropin releasing hormone and luteinizing hormone release, gonadotropin releasing hormone is also referred to as luteinizing hormone releasing hormone. So the gonadotropin releasing hormone is secreted in pulses from the hypothalamus, act on anterior pituitary to stimulate the luteinizing hormone and follicle stimulating hormone release, with luteinizing hormone mirroring the pulses for gonadotropin releasing hormone more directly, while follicle stimulating hormone respond in a slower and modulated manner. Both the luteinizing hormone and follicle stimulating hormone are secreted by specialized cells in the anterior pituitary gland known as gonadotrophs, and their secretion is highly dependent on gonadotropin releasing hormone that comes from the hypothalamus. If this there is no gonadotropin releasing hormones, so there will be no luteinizing hormone or follicle stimulating hormone. So the luteinizing hormone and follicle stimulating hormone are glycoprotein hormones. They are composed of alpha and beta subunits, with beta subunit conferring specificity. Luteinizing hormone and follicle stimulating hormone bind to their respective receptor on testicular target cells. Luteinizing hormone binds with the Leydig cell and stimulates testosterone production, while follicle stimulating hormone binds to Sertoli cell and it stimulates spermatogenesis and produce inhibin. Both hormones act through the cyclic AMP, cyclic adenosine monophosphate second messenger system, which activate protein kinases and regulate enzyme subsystems within the cell. Ultimately, drive synthesis and secretion of hormones or proteins necessary for reproduction. So, to summarize, these hormones, luteinizing and follicle stimulating hormone, are glycoprotein gonadotropins that are secreted by anti-appreciatory gonadotropes under the control of gonadotropin releasing hormone, and they act on Leydig and Sertoli cell through cyclic AMP second messenger pathway to regulate testosterone secretion, uh, spermatogenesis, and secretion of inhibin. So testosterone is produced by the interstitial cells of the Leydig intestines that require the stimulation by luteinizing hormone from the anterior pituitary. The increase in LH will increase the production of testosterone. So this luteinizing hormone bind to the receptor on the Leydig cells and then activate the cyclic AMP protein kinase uh, pathway and stimulate the secretion of testosterone. So these secretions are directly proportions. Testosterone and luteinizing hormone secretion are directly proportioned. Then these Leydig cells in the early infancy, the mature Leydig cells are present for a few weeks after birth. In the childhood, these Leydig cells regress and disappear. The testes contain only immature fibroblast-like precursor cells, and at puberty, rising luteinizing hormone secretion causes these fibroblast-like cells to differentiate into functional Leydig cell, which begin production of large amount of testosterone. So injection of the purified luteinizing hormone in a child can induce the differentiation of fibroblast-like precursor into active testosterone secreting Leydig cell, that is confirmed by experimental studies. So the key regulator of this mechanism is luteinizing hormone. It is not this luteinizing hormone not only stimulates the testosterone secretion in the mature Leydig cell, but also induces the development and maturation of Leydig cell at puberty or experimentally at any age. Then luteinizing hormone, when it is secreted, it stimulates the secretion of testosterone. Once produced, we have seen that these secretions are directly proportional. Once this testosterone produced, it acts in reciprocal or inhibitory manners on the hypothalamic pituitary axis, and it inhibits the hypothalamus. This reduces the gonadotropin releasing hormone secretion, and lower GnRH will result in decreased luteinizing hormone and follicle stimulating hormone secretion from the anterior pituitary. So there will be reduced luteinizing hormone. In, so with less stimulation, testosterone secretion by the Leydig cell then will decrease. So if testosterone rises too high, there is increased inhibition. That means reduced GnRH, LH, and FSH. So testosterone level falls back to the normal. If testosterone drops too low, the hypothalamus increases the secretion of gonadotropin releasing hormone that act on gonadotropes in the anterior pituitary, and then these pituitary cells they release luteinizing hormone and follicle stimulating hormone. The testes produce more testosterone. So this is a classic negative feedback loop that keeps testosterone level within a narrow physiological range. This system ensures stability of male sexual function, spermatogenesis, and androgenic effect without overproduction. In short, the testosterone secretion is self-regulated through negative feedback at both the hypothalamus and the anterior pituitary. This maintains hormonal balance. The follicle stimulating hormone binds to its receptor on Sertoli cell that is located uh, the Sertoli cell that is located on the seminiferous tubule. This stimulates Sertoli cell to grow and mature and secrete spermatogenic substances including nutrients and signaling molecules that support developing sperm cells. Sertoli cell can also produce inhibin which provide negative feedback on follicle stimulating hormone. So testosterone that is secreted by Leydig cell in the interstitial spaces diffuses into these seminiferous tubules along with its potent metabolite dihydrotestosterone. It exerts a tropic effect on spermatogenesis by promoting the differentiation of spermatogonia, supporting meiosis and maturation into spermatids and spermatozoa. And there is also synergistic action. Both FSH and testosterone are required together for the initiation and maintenance of spermatogenesis. FSH prepares and activates these Sertoli cells, and testosterone and dihydroxytestosterone they directly stimulate the germ cell to complete the development. So we can summarize that follicle stimulating hormone act indirectly through Sertoli cell, and testosterone they act directly on germ cell within the seminiferous tubule. Their combined effect is essential for normal spermatogenesis. Now, what is the role of inhibin in this negative feedback control of seminiferous tubule activity? When the sperm production is low, the pituitary follicle stimulating hormone secretion is increased, while the excessive spermatogenesis will cause this follicle stimulating hormone from the anterior pituitary to decrease. This fluctuation is controlled by inhibin, that is a hormone secreted by Sertoli cell in the seminiferous tubule. This is a glycoprotein hormone with a molecular weight of 10 to 30 K. It has been isolated directly from cultured Sertoli cells. The inhibin directly acts on anterior pituitary gland to suppress the secretion of follicle stimulating hormone or FSH, and this effect is specific to follicle stimulating hormone. Unlike testosterone, which inhibit both the luteinizing hormone and follicle stimulating hormone via hypothalamic gonadotropin releasing hormone suppression. So, what is the physiologic importance of this? It provides an important negative feedback loop. It prevents excessive stimulation of spermatogenesis by follicle stimulating hormone and ensures a balanced rate of sperm production. And this works in parallel with testosterone's negative feedback on gonadotropin releasing hormone and luteinizing hormone. Together, these two mechanisms, they tightly control or regulate male reproductive function. So the inhibin that is secreted by Sertoli cell is key regulator of spermatogenesis by directly inhibiting follicle stimulating hormone from the anterior pituitary. It prevents the overproduction or excessive production of sperm and work alongside testosterone feedback to maintain the reproductive homeostasis. Human chorionic gonadotropin is secreted by the placenta during pregnancy. This is secreted by the placenta during pregnancy and enters the circulation of both the mother and the fetus. HCG is a glycoprotein like luteinizing hormone, and it has got a functional and structural similarity to luteinizing hormone. It binds to the luteinizing hormone receptor and mimics its action on target tissue. So in the male fetus, HCG stimulates fetal testes or Leydig cell in the testes. This results in secretions of testosterone during intrauterine life. Fetal testosterone is critical for male sexual differentiation, including development of internal genitalia. Fallopian duct will give rise to epididymis, vas deferens, and seminal vesicles, and development of male external genitalia, that are the penis and scrotum. And this is also important for the descent of the testes later in gestation, which helps from with the help from androgens and insulin-like peptides. So without HCG stimulation, the fetal testes would not produce adequate testosterone, and deficiency in this pathway may lead to ambiguous genitalia or failure of male sexual differentiation. So to summarize, we can say that during pregnancy, placental human chorionic gonadotropin hormone or SCG, beta SCG, act like luteinizing hormone, and it stimulates fetal Leydig cell to produce testosterone, which is essential for the development of male reproductive organ.
Thank you for watching this video. If you have any query, please write in the comment box.